everyone can please sign in on this attendance sheet. We'll just wait a few more minutes.
Yeah, just maybe a couple more minutes. I'm looking for the students.
There's gonna be some late stragglers.
Carolyn.
I think it's Carolyn.
Spelled like Carolyn.
Okay.
Okay. Yeah, my
Okay,
parents were
You
just
can
like,
hear
you
us
know
over
what?
here. Dang it. It's so loud.
Maybe I will start. Yeah, maybe. Wait. I'm supposed to take that. But I want to say. My family is refugees in Europe, like my mom and sister. They want to go back.
Which is true.
Yeah, that's been a year.
Oh, no, this isn't really grad school. Work. Yeah. This life is like it's not grad school. Yeah, it's the same. I had a bad name out of the whole time. I was going to say, anyways, we'll just pay you.
Your wife's family, yes. Yeah, that was. Huh? My mom was urged her to leave. She lived in the world. She was not. But they moved really fast. The first struggle was outside the trip. Yeah. Just 1 moment. Yes, things are going well.
All right.
You got promoted and this year your papers was
Okay, it looks like that.
Okay, I think I will start introducing this seminar series. So these are seminars from Genome Science Institute, or GSI. I'm GSI co-director, Alla Grishok. This is GSI director, Nelson Lau. Rachel Flinders, another GSI director. And where's Abby? Abby Gatanti is our administrative associate. And if you want to receive information about our events, you can sign up to basically email GSI at bu.edu and ask to be GSI member, and you will get emails about our events. They're also advertised, with you can see on boards and electronic advertisement. We usually have seminars on Tuesdays. It's not set in stone which Tuesday. We try to do it once a month, but now we actually have seminars more often, and I hope you will be interested in seeing more of seminars that are actually a lot of them are focused on technology development. So today we'll have somebody introducing, but September 26th will be Sarah Rohanifard talking about developing tools for single molecule sequencing and imaging of RNA modifications. On October 3rd, we will have a talk from Professor Winston Timp, who is using nanopore technology for sequencing, followed by Oxford nanopore technology seminar. And October 17th, we'll have Rachel O'Neill from University of Connecticut, who will be talking telomere to telomere scale genomics for study centromere function and evolution. But today's seminar is hosted by Professor Yuri Alexeyev, who is director of microarray sequencing service facility and single cell core facility. And Alexeyev will be introducing our speakers.
Thanks, Ola. Thanks to GSI for organizing this technology series seminar. Thanks to Abby for great advertisement, I guess. And welcome everybody, thanks for coming. So today we're hosting 10x Genomics. This company, we started using their products in 2017 when Department of Medicine established the single cell sequencing core facility. And overall, this was a very successful journey. Believe it or not, we processed for single RNA-seq, we processed over 1,500 samples relatively successfully. However, I cannot say that it was always spotless. There are a couple bottlenecks that one related to pricing, which was ridiculously high compared to like at least 5 times higher than to make a bulk RNA-seq library, and even more so technical bottlenecks, because to get a successful data you need suspension of viable cells, ideally 90% viability, and when you're dealing with real life samples, it's not always easy to get that. And if you have frozen tissue, the only choice was to do single nuclei sequencing, and single nuclei extraction is also not so easy to do sometimes, depending on the type of sample and things like that. So fortunately, 10x Genomics consistently improving their products and protocols, and recently this kit was set up, which is called Gene Expression Flex. That allows. Sorry?
We can't really hear you in the back, Yuri.
Oh, I'm sorry. Should I start over?
Yeah, please.
Yeah, no, I was talking about the bottlenecks that we had with previous versions of 10x. Sorry, it's too loud now. So I was saying about bottlenecks, first of all, pricing of the previous kits, and also that technically it was not always easy to generate a 90% viable cell suspension for successful experiments. And if you have frozen tissue, you had to do nuclei extraction, and that's also not always straightforward. So fortunately, 10x is improving their protocols and products consistently, and now we have this Gene Expression Flex kit, which allows you to work with frozen and fixed samples, and even FFPE, which was unthinkable just a couple of years ago. And this is what I hope this seminar will be focused on. So it's my pleasure to introduce our today's speaker, Carolyn Peterson, who is Science and Technology Advisor at 10x Genomics. Prior to that, she got a master's degree from Tufts and worked for a long time in Broad Institute, so she's an expert in single cell and next generation sequencing. Carolyn?
I'll just get set up here. Thank you for that wonderful introduction. You already made a lot of great points that I was going to touch upon today, so half the talk's already over. So I'm Carolyn. I'm your Science and Technology Advisor from 10x Genomics. If you can't hear me in the back, just raise your hand and I'll just adjust the mic. Yeah, so today I'm here to talk about our new assay developments in terms of single cell. So yeah, sorry, rough start there. But yeah, so today I am here to talk about our single cell flex assay. It does allow you to run fixed or FFPE samples. So at 10x, we started as a single cell company. We have expanded into the spatial world. I do want to just, before going too much into the weeds with the Flex assay, I want to give you sort of a 30,000 foot overview of the different platforms we have to offer. All three of them can run FFPE. We designed them to be complementary to each other, so you can actually run all three of our platforms from the same FFPE block to really, tackle your biological questions from different angles. We'll then zoom in a little bit on the Chromium single cell platform, more of a 10,000 foot view. Just going to go over all the different assays that we have to offer. We have a lot of different options for different, multi-omic views of the individual cell. And then we're going to really zoom in and do a deep dive into the single cell gene expression flex assay. How it works, how well it works, kinds of samples you can use with it. And then finally, I'm going to introduce some application highlights. It's a pretty new product, but it's gaining some, increasing popularity. And, the preprints are just starting to roll out. So I have two good ones that I'm going to dive into. And then finally, I'll take some questions. We have some time. We'll have some time left over. I think after that, we'll convene for lunch and then, some more discussions afterwards. So it goes without saying that the complexity of biology is immense. Trillions of cells within your body, millions and millions of cell-to-cell interactions, that are synchronized and orchestrated with each other to create those complex tissues and organs. To really understand the microenvironment of these tissues down to the molecular level, you want to be able to measure the gene expression, at the single cell level across different tissues, but also within individual cells. So at 10x, we've developed, a real arsenal of tools that you can use to tackle those complex biological questions from multiple different perspectives. So of course, we have the Chromium single cell platform. You get your gene expression measurements at the individual cell level. Very rich data sets. With bulk RNA sequencing, you do get gene expression measurements, but it's for an average gene expression measurement for all the different cell types within your sample. If you're looking at complex tissues or even tumor development, you want to understand the different cell populations that are present and how they're interacting with each other. So yes, it can be a little bit expensive, but you're getting a lot more information. With our new Flex assay, you can now get beautiful data sets from FFPE. I used to do a lot of bulk RNA sequencing at the Broad on FFPE. For some rough data sets. But I'll show you today, we're really able to create these in-depth, powerful data sets that allow you to get a deeper understanding of your tissue or disease of interest. Now, single cell is great. You get very high resolution gene expression measurements down at the single cell level. But you have to dissociate your cells into a suspension. And you lose an important context, and that's the spatial context. You want to understand what genes are being expressed, but especially with tumor development, or in this case, mouse embryo development, you also want to know where those genes are being expressed. So Visium Spatial is basically like just sequencing an intact piece of tissue. It uses a gene expression slide that you put the tissue on, and then it captures the transcripts from that intact piece of tissue, so you can see where those genes are being expressed. We do have a brand new platform called Xenium in situ. So this actually is a microscopy-based readout rather than sequencing. It's a more zoomed-in, targeted approach. Chromium and Visium are whole transcriptome approaches. They're great discovery tools for generating hypotheses. Xenium is for that really, zoomed-in approach into your tissue and to test those hypotheses. You have a subset, a pre-designed panel of a couple hundred or more targets of specific genes, but you can see where they're being expressed within your individual cell, within the nucleus, within the cytoplasm. So really, there's multiple different ways of interrogating your samples, and there's multiple different resolutions. Xenium is by far our highest resolution yet. So all three of these platforms, with the release of Gene Expression Flex, are capable of running FFPE. This preprint that came out recently, I'm just going to do a quick highlight overview, but it did use, the same FFPE tissue block, took serial sections, ran it through 3 different platforms. So I used Gene Expression Flex to get that single cell resolution, capture the heterogeneity of that tumor sample, and annotate the different cell populations present. With Visium Spatial, they were able to map where those cell populations were present and see how they're interacting with each other within the tumor microenvironment. Now, as I mentioned, Chromium and Visium are whole transcriptome approaches. What's really nice, and we did this on purpose, is that we use the same probe-based approach. So you're able to easily integrate your data sets together and switch between the two data sets. You can further deconvolute, the Visium resolution to get even more deeper insights into your tissue of interest. With Xenium, they use a pre-designed panel for the particular tissue type of interest. It's about 300 or so genes, but they were able to get extremely high resolution into that tumor microenvironment. With Chromium and Visium, they captured the full heterogeneity of their samples. Because of the shared probe designs, they were able to integrate their single cell data with their spatial data and map out where those cell populations, how they were located within the tumor microenvironment. With Xenium, they got that targeted zoomed in approach for an even closer look at what's going on inside that tissue. And one thing I did want to highlight from this paper is what they discovered with Xenium. So because they had such a high resolution, this tissue was originally annotated as double positive, but they found this very small section of tissue that was triple positive. Now this was only discovered, when they looked at the Xenium data at the really high resolution. It's a very, very small spot within the tissue section. We went back to the Chromium and Visium data. That spot was there. It was just overlooked because it made up such a small amount of the overall tissue. So, for reference, that capture area on that Visium slide, it's about 5,000 spots. This particular spot only took up 6 spots. So Xenium, provided that resolution to be able to identify that spot, but then they were able to go back to the Visium and Chromium data, go back to that whole transcriptome data set, and they could perform differential gene expression between those 6 spots and the remaining 5,000 or so spots. And they were able to identify almost 100 genes for further follow-up. This really just highlights our FFPE capabilities across the board. You have a very powerful toolkit. Whichever assay you use, you're going to get very rich data sets and very confident results. It's going to be even more powerful if you combine different platforms together. You're going to get different angles of your overall biological picture. But today we're here to talk about Chromium single cell. Now, again, biology is very complicated, even at the individual cell level. Chromium single cell has been out for a bit of time. We've come out with a lot of different assays to really address the multi-omic needs. There's a lot more going on within your individual cell beyond just gene expression. If you are interested in just the mRNA transcripts, the overall gene expression, you can use our 3' or 5' assays. In terms of sensitivity, they're both on par. The difference is just which end of the transcript you're capturing, 3' or the 5' end. With 5', you do have the added bonus of being able to capture VDJ transcripts. So you can characterize T cell or B cell receptors and look at the immune system, how it's responding to a particular treatment or progression of disease. Now we do have some add-ons called feature barcoding that enables that, those additional analytes to be measured. You can perform proteomic studies, look at cell surface protein expression alongside gene expression. You can also do functional genomic studies and run single cell CRISPR screens. We have another add-on, another feature barcoding add-on called BEAM. It's really targeted towards immunologists. So it allows you to do high throughput antigen specificity screening. So you'll get your gene information, VDJ information, for your TCRs and BCRs, but now you know which antigen they're actually specific to. If you're interested in epigenetics, we have a multi-omic solution that combines RNA-seq and ATAC-seq data from the same single cell. So you can look at the upstream epigenetic regulation and how it's affecting that downstream gene expression. Today's main focus is our newest assay, Gene Expression Flex. It was originally designed for 4% PFA fixation. It has been shown to work beautifully on FFPE samples. So that's what we're going to dive into today. As you can see, it's really, the Chromium platform is really a Swiss Army knife in terms of what you want to look at in terms of single cell. I could spend the full 2 hours talking about the whole portfolio, but I'm not. We're just going to look at Flex. Although we do have a lot of different assays, the overall workflow is more or less the same. You know, we aim for a streamlined, simplified workflow to make it as user-friendly as possible. So in general, you begin with a suspension of isolated cells or nuclei, and then you load it onto the Chromium X instrument. This is the real key behind our technology. So this creates those single-cell droplets that give you that high resolution. After you lyse open your cells, capture your transcripts, produce barcoded cDNA. It's a pretty standard library construction protocol. After sequencing on your next generation sequencer of choice, the protocols we have are compatible with Illumina short read sequencing. You can do long reads, unfortunately not for FFPE, and I'll show a little bit why in a little bit. But it is a full end-to-end solution. We provide our cell ranger analysis pipeline for analyzing your data, and then you also have a secondary visualization software for your secondary analysis. So here you can look at your t-SNE or UMAP plots and annotate the different cell types present and perform that differential gene expression. This is a little bit of a closer look at what goes on inside the Chromium instrument. I apologize if this is redundant for any of you. Technology's been out for a bit of time. But in general, you'll load your cells, your necessary reagents, and then these barcoded gel beads into the Chromium instrument. They flow through tiny microfluidic tubes and are partitioned off in an oil and emulsion droplet, containing a single gel bead and a single cell. We call these gems for gel bead and emulsion. Now within each gem, you're going to lyse open your cells. The transcripts will be released and then captured by the oligos on those gel beads. Reverse transcription will create uniquely barcoded cDNA, after which you can break open those gems, pull all the molecules together. Pretty standard library construction protocol from there. In terms of overall throughput, you can run 8 samples at a time, 10,000 cells per sample. We have a capture efficiency of 65%. So you do kind of want to overshoot maybe 16 or 17,000 in order to get that 10,000. We have a multiplier rate of 0.8% per 1,000 cells. That's the rate at which you have more than one cell per gem droplet, in which case you lose that single cell resolution. So that is important, especially when you're increasing the overall throughput. And I'll touch upon that when we talk about multiplexing for the flex assay. In terms of the gel beads, the oligos across the gel beads are more or less the same. You're going to have your sequencing primer, your unique 10x barcode. So that's unique for each gel bead. So all the transcripts from a single cell will have the same 10x barcode. And that's how you do that single cell demultiplexing. Unique molecular identifier, counting the unique transcripts, making sure you're not counting PCR duplicates and just getting that overall gene expression information. And then the capture sequence. So that is going to change based on which assay you're going to run. With our standard 3' gene expression, it's that familiar poly-DT tail. It's going to capture the mRNA by the 3' poly A end. Now with Flex, instead of capturing the transcripts themselves, mostly because when you fix your samples or work with FFPE, your transcript's going to get very degraded. And you might not even be attached to that poly A molecule. So that traditional 3' capture is not going to give you a lot of information. So we actually use a targeted probe-based approach. The probes themselves will have a complementary capture sequence built within them so that they can be captured directly by those gel beads. So that was the really quick crash course and Chromium overview. But let's dive into gene expression flex. Now, it used to be called fixed RNA profiling. We rebranded it to flex because of the increased flexibility. We are very creative at 10x. So, as Yuri mentioned, working with the other assays, you can do fresh or frozen samples. You really want a very high level of viability. 70% works. We recommend 90% or higher. As soon as you dissociate your cells, they start dying. And if you're working with a lot of cells, you're really in a time crunch. Also makes it very difficult to ship your samples to another lab if you don't have a port. Makes it difficult to acquire samples from other locations. Now, when you fix your samples, you're locking in the biological states. You can wait and store them. When you run fresh samples, you're usually only going to do 1 or 2 at a time because of that time crunch. With fixed samples, you can wait and run all 8 at a time. If you want a multiplex, if you have a lot of samples, you can do 128 samples in a single run and 1 million cells per chip. That's a lot. So we do have multiplexing methods for maximum flexibility for your throughput options. It does also make it a lot easier to ship your samples and get samples from other locations, such as FFPE samples. You can just get a couple of scrolls from someone at an FFPE block and have that shipped to you. So with gene expression flex, it's really any sample at any scale. Fresh or frozen tissue, you can fix with 4% PFA fixation method. Same goes for cell suspension or isolated nuclei. And we do have have shown that we are very compatible with FFPE tissue as well. This is excellent because, especially in a clinical setting, sometimes FFPE is really the only kind of sample you can get your hands on. There's a lot of FFPE samples that have, they're a notoriously challenging sample type. Prior to Flex, it was hard to get really good data from FFPE samples. There's a lot of FFPE kind of just sitting around that might be like a decade old. But now you can run it through single cell Flex and still get really good results, even though it's a decade or older. So because of that high degradation that happens with fixation, FFPE preservation, those mRNA transcripts are going to be very highly degraded. So we use a targeted probe-based approach. So there's actually going to be two sides of the probe, left-hand and right-hand side. They both have to hybridize to the mRNA target, and there has to be a perfect match for them to ligate together. That's going to actually improve our specificity and off-target binding. This blue section here is that complementary capture sequence. So the probe gets captured by the gel beads after it ligates together. There's also a built-in sample-specific barcode that's going to enable multiplexing. In terms of throughput, you can do the single-plex assay. That's just one sample per channel. You can do 4-plex, 4 samples per channel, and then 16-plex. That's going to give you that 128 per run and 1 million cells per chip. When you do that high of multiplexing, with single-plex, you target 10,000 cells per sample. With multiplex, you still want to get like 8 to 10,000 cells per sample. If you run 10,000 cells total divided by 16, you're really not going to get a good data set. So you increase your cellular throughput. You can go up to 128,000 cells per channel. That's what gives you that 1 million cells per chip. Now that multiplet rate that I mentioned before, 0.8% per 1,000 cells, it's going to rise when you get up to 128,000. However, because, and I'll touch upon this in a later slide, but because you have that sample-specific barcode with multiplexing, you're actually able to demultiplex any doublets that are formed as long as the cells come from 2 different samples. So you can create very large data sets. You're also creating more usable data. You're not throwing out all of those reads. So let's take a closer look at the actual probe design and the overall workflow. For probe panels, we have human and mouse available. It's a whole transcriptome approach, 18,000 genes in human, 20,000 in mice. We do have the exact gene information on our website. If you're interested in what the panels include, we can certainly send you that information. We do also have options for custom probes. If there is a gene of interest, that's not included. Now, in terms of the probes themselves, they only target, that target site is only 50 base pairs long. So that's really good for degraded samples. You don't have to capture that whole transcript. In terms, I already talked about the specificity. Both of these left-hand and right-hand sides have to hybridize. There has to be a perfect match here in order for that to ligate together to form a consecutive molecule. If there's only one side of the probe that ends up being captured, it's not going to extend. It's just not going to end up being sequenced. In terms of sensitivity, we've actually shown that this is the most sensitive assay that we have to date, more than 3'. We designed our panel so that every annotated isoform of every gene has a 3-probe coverage for that maximum sensitivity. Of course, we validated these panels against 3' gene expression. We see excellent correlation when we look at the gene expression profiles between 3' and fixed RNA. This is really, what this is telling you is that your biological insights are going to remain consistent. Now what's really nice, when we look at the sequencing saturation curves, you'll actually notice, we're looking at the median genes detected per cell as a function of reads per cell. The flex assay is actually more sensitive. You're detecting more genes at a lower read depth. So this actually reduces your sequencing burden by half. So even if you're not multiplexing, you're gonna see savings in sequencing. With our standard assays, we recommend 20,000 read pairs per cell at a minimum. With 10,000 cells per channel and 8 samples at once, that's a lot of reads. It's gonna be cut in half with Flex. It's only 10,000 read pairs per cell because we have such a targeted, sensitive, probe-based approach. The workflow, as you'll notice, very similar to our other workflows. We try to keep it as simple as possible. The main difference is, if you're starting with fresh or frozen samples, you're gonna fix them first. And then when you're ready to run, you'll permeabilize your cells and hybridize those probes so they can go within the cells and hybridize the mRNA targets. You'll load it onto the Chromium instrument, and again, the probes themselves are captured rather than those highly degraded RNA transcripts. We do have feature barcoding available for Flex if you are interested in proteomic studies. It does require you to start with fresh samples. You'll do your antibody staining and then fix them. But in terms of the workflow, you'll just create a second library and then you can sequence them together. Cell Ranger will take care of the rest for you. I do want to mention that a library construction protocol is even simpler, which is great if you're the one doing it in the lab. Since we just have those short probes, it's just really a simple SI-PCR step. When you're dealing with the full-length transcripts, you have to do fragmentation, end repair, it's a bit longer. So again, even more streamlined workflow. Now with multiplexing, that's where that sample-specific barcode comes in. So the general idea, in this case, we're using 4 samples. We're using our 4-plex solution. So you'll have 4 different samples, 4 different probe panels. Now these panels, the targets are the same. You're targeting the same exact gene. The only difference is that sample-specific barcode that's built in. So you'll take your 4 samples, hybridize each probe panel to each, and then you can pool them together and run them on the Chromium instrument. So that's going to really increase your cost savings. If you're using 1 reaction out of your kit instead of 4, or alternatively instead of 16. So you really see significant cost savings when you do multiplexing with that decreased sequencing burden. One other benefit of multiplexing, when you pool your samples together, you're just going to do like a pooled wash. So your cell loss is actually going to be minimized. So with multiplexing, you can actually have a lower input into the assay than with singleplex. For FFPE, we recommend for singleplex, it's like 400,000 cells. When you're multiplexing though, it's 100,000, 200. It's lower.
Yeah.
It's lower. It's better. And then of course, Cell Ranger is able to demultiplex that data for you.
That's a good question. Can you go back a slide? In this flex approach, do you still need a single cell suspension, or because it's fixed, you can't?
Yeah, so you do have to suspend it. You can't just throw solid tissue into the instrument. It's going to cause some clogs. You do dissociate, so you'll start with the FFP tissue sections and you'll dissociate that. We have a demonstrated protocol available. You get whole cells from FFPE. There's also protocols out there for, and I'll show these later, for isolating nuclei from FFPE. In terms of fixing your samples, if you're doing the 4% PFA fixation method, you have a couple of options. If you're starting with tissue, you could dissociate and then fix them. You could chop it up and then fix it and then dissociate that. That's actually going to work better for fragile cells. That's been shown to help preserve fragile cell types. So as you'll see in the subsequent publications I show, depending on how you handle your samples, the overall cell populations are going to be detected, but they might be in more or less abundance depending on how they've been processed.
So it's not that the hybridized probes, after they get washed out, the cells, you still have to get a cell suspension.
Yeah, so you'll do your cell suspension first, and then you'll do the probe hybridization.
Okay.
Yep.
What kind of fragile cells are you using for fixed tissue?
Oh, off the top of my head, let me look into that for you. I can't recall off the top of my head. I know I have a slide somewhere that discusses it, but let me follow up with that for you. So, in terms of multiplexing, we've of course done extensive validations just to make sure that multiplexing your samples isn't going to decrease your overall data quality. So here we took a Single sample, we had a lot of sample that we could split into 16 aliquots. Added the 16 different probe panels to each aliquot and then pooled them all together. Basically, we see that these cell clusters are consistent. There's no dropouts in particular cell populations. I do like to point this out because, if you have a lot of a particular sample, or if you really want to detect rare cells and you need a higher throughput, you can just take one sample and increase the throughput for that one sample and go up to 128,000 cells. Now, you might be wondering, why do you want to multiplex to begin with? Why can't I just do 128,000 cells for one sample? The answer is that doublet rate. When you get to 128,000 cells, your doublet rate's over 6%. Now, with flex, as long as your doublets are coming from two different sample types, they're going to have the probes will have their own unique cell barcode. So Cell Ranger can not only detect the doublets, but you can also demultiplex them. So you're not throwing away 6% of your reads. So again, more data and more usable data. In terms, going back to the workflow really quick, you do have multiple safe stopping points. You can stop after fixation or after hybridization and pick up whenever you need to. We've done a lot of validations looking at different stopping points, different tissue types, storage conditions, and duration. Longer term studies are underway. It just takes a while. So we've tested up to 6 months so far. In terms of the sequencing results, very consistent results. Your quality is not impaired with your different storage conditions. More importantly, your cell populations are not impaired either. They're still present, in the same overall abundance. Sample compatibility. So again, we have human and mouse panels available. If you want, if you're interested in other species, definitely let us know. Flex is becoming a very popular product, and we're working to expand this panel, but we really want to build what the customer needs most. We do take customer feedback very seriously. Every request is logged. If there are species that you want to see, definitely let us know. If you're working with non-human primate, a lot of people use the human panel with really good results. In terms of samples, cell lines or isolated nuclei, fresh tissue, frozen tissue, you can just fix that with a 4% PFA fixation method. You can also run FFPE, so that's a pretty new capability. We see really beautiful results. So of course, this is just a screenshot of one particular sample. What you really want to see here is the distinct cell clusters and the different cell populations that are all detected. You'll see a good separation between the cells detected and that background signal. And most importantly, your library complexity is high. So you get high UMIs detected per cell and high genes per cell. Again, we have that demonstrated protocol for dissociating FFPE tissue. Two different options. You could do it totally manually or use Miltenyi products. What's nice about the protocol is that they include all the tissues that they tested, how much tissue they use, 25 or 50 microns, two scrolls, tissue sections, 25 or 50 each. Generally, we recommend doing 50 for mice, but you'll also see the amount of cells that were obtained. So if you're working, with mouse tissue and you might not have enough cells, you can know ahead of time how many mice you might want to pull together to get enough of a cell recovery. Again, we also have that custom probe design. So this is a lot of information, but basically it just gives you all of the information that you need for designing your own probes. What kind of sequences you want to avoid, the GC content. So you can really just purchase or create your own probe, order it, and you spike that into the panel mix. So I've seen people do that. There's been a recent group that has been doing CRISPR screens with fixed samples. They just create custom probes for their guides. So they're capturing the transcript information as well as the guide information. You can also do it, maybe for viral genes that's not included in that original transcript. So again, a lot of flexibility, a lot of options. We've tested how the fixation protocols differ between 4% PFA fixation and FFPE. The clusters correlate extremely well with each other. Again, that sample preservation and processing is, it might affect some cell populations more than others. The main takeaway, there is a strong concordance between the majority of the populations. None of these populations are completely missed. So, either if you're doing 4% PFA fixation or FPE, you're not just going to eliminate a cell population and completely miss that from your data sets. So we have tested a lot of FFPE in-house. Tests are still ongoing. We've done human and mouse, healthy and disease, multiple tissue types. We have a lot of data sets on our website. We have some secret data sets too. They are customer facing, they are public, but we don't put them on our website because we just have so many. So if there's a tissue of interest that you would like to get more information on, definitely let me know. Because we might just have a data set for your specific tissue of interest, for FFPE. So we can certainly dig around, find out as much information as possible. There's a lot of resources that aren't just, on our website because we have a lot. But there's a lot of data sets available so far. If you are interested in what the data looks like, you can download the data sets for free. The software is completely free, so you can just, play around with the data sets and see how it looks. So to wrap up, good on time. Yeah. I'm going to talk about 2 application highlights. The first one, really high level. This one came out of Berlin. So they took actual patient samples, lung tumor samples, and split it into 3. They ran a fresh sample through 3' gene expression. And this is the freshest of the fresh. They just, took the sample, put it on ice for 30 minutes, and then ran it. That's not, that's really ideal. For this paper, it's great because they have really high quality results to compare to. It's not always going to happen in the real world. You're not just going to get a sample straight from a biopsy and go run it on 10x. It's going to be a little tough. They also did cryopreservation, stored it for 4 to 5 months, and then ran it. Did nuclei isolation because, starting from a frozen sample, nuclei isolation does sometimes work better because you take a big hit on cell viability when you thaw your samples. So they ran single nuclei isolation on the 3' kit. Finally, they did FFPE preservation, started at 4 to 5 months at room temperature, and then ran it through Flex. So here, I do want to point out they ran single nuclei isolation. Our protocol, is for whole cells. But the first people that did FFPE with the Flex kit, which is the next paper I'll show, actually, it's called SN Pathoseq. They actually did nuclei isolation. This paper came out in April, so the work was done before that. It came out before our dissociation protocol came out. So they just followed what was publicly available so far. Just a quick highlight, overview of what they found. When they looked at the 3 data sets, they made a combined UMAP, and the different cell clusters, were well represented by the 3 different data sets. So you're not really missing any cell populations. The cryopreserved samples, the overall quality wasn't as great. They didn't really see a great, cell-to-cell mapping between fresh and cryopreserved and FFPE. A couple of reasons why that might be, freezing and thawing takes a hit on your overall quality. And nuclei, you're looking at pre-spliced mRNA, so it's, a little bit different. Overall, your biological insights are going to remain consistent, might just not be as much of an apples-to-apples comparison. So because that cryopreserved sample wasn't as great, they really just wanted to look at how that really, really fresh sample compared to that FFPE sample. So, the best of the best versus a FFPE sample that's the transcripts are very degraded. We're seeing the same thing that we saw before. Those clusters are very well correlated with each other. The overall cell type proportions are going to differ based on how you process your samples. So, it's not the same exact protocol. You're handling your samples differently. There's going to be slight differences in the abundance of cell populations. However, those overall cell populations are still detected. They also looked at, this is just a handful of gene signatures, but they looked at the correlation of, gene signatures and how much they were expressed between FFPE and fresh tissue. Again, they see excellent correlation. And in their own words, they said that, specific and clinically relevant features of cancer are robustly preserved in single-cell FFPE data. And you can use single cell FPE to replace 3' gene expression with fresh tissue. So that's great. You know, you aren't constrained by fresh samples anymore. What's really great is you can run retrospective studies for FP samples that have been sitting around for a while. And what else is great is you also have clinical information from your samples. You know, if it's been a couple of years, you probably know how the disease progressed, how they responded to treatment, so you can directly correlate your single cell results immediately to the clinical phenotypes. Now, this is the paper I did want to highlight a little bit more. So this is the S-Palpacy paper. They were one of the first ones to do FFPE with Flex. First thing they did, though, was validate how well it worked. So here they're looking at the 4% PFA fixation method versus fresh. So not FFPE versus fresh. Just because they wanted, this was before we came out with our FFPE compatibility. They just wanted to see how it actually compared in a head-to-head comparison. You know, maybe they didn't want to take our word for it, so they just wanted to do it themselves. What they found is that the Flex assay was in fact more sensitive. You see higher levels of UMI counts per cell and higher levels of genes detected per cell. Also much better detection of lowly expressed transcripts, such as these transcription factors FOXA1 and TP53. You know, in orange you see the fresh sample results for 3'. They're barely detected. With that targeted probe approach, we're getting much better coverage of those lowly expressed genes. They also see more refined cell clustering. So here they're clustering based on the cell phase. You can see a much more defined cluster for those S phase cells, which again, you know, more powerful insights into their data sets. They're better able to tease apart those different cell populations present. So once they, you know, confirmed that Flex did in fact work as well as we said it did, they went ahead and, you know, saw how well it worked on FFPE. Now they took a very challenging sample to work with, which was, you know, bold considering it was a proof of concept study. The sample was a liver metastasis breast cancer sample. Maybe switching those around. But it was taken during an autopsy 4 hours after the patient's death. So the RNA was expected to be extremely degraded. And what they did is they actually did a nuclei extraction from FFPE. They developed their own protocol. It's the same protocol as that previous paper used. And even though they chose, you know, a very challenging sample type, they got really beautiful results with their FFPE sample. Again, really distinct cell clusters here. But they did notice that was almost a complete lack of immune cells. So they were worried, you know, maybe their FFPE protocol just depleted their cell populations, or maybe, you know, this tissue type was known to be an immune cell desert, so maybe they just weren't there to begin with. In terms of complexity, high number of genes detected per cell. You know, you're getting rich data sets. And what they found that was really interesting, so this particular tissue sample was supposed to be triple negative based on clinical evaluation. They found the expression of estrogen receptor 1, BSR1, which is totally missed by clinical validation. Again, you know, is this a fluke? So what they did is they compared it with our Visium FFPE. So this was already an established protocol. They knew it worked. They wanted to see how well their FFPE results compared to Visium FFPE. Again, those probe sets that I described are the same between single cell and Visium. So they were able to integrate their data sets together and ensure that they were having, you know, real apples to apples comparison. Gene expression profiles, you know, were beautifully correlated with each other. The overall cell population, slight differences, but again, great concordance. They again found the lack of immune cells, so it did confirm that it wasn't a kind of an immune cell desert, and they weren't just completely destroying their immune cells with this protocol. And because of those shared probe designs, they were actually able to integrate their single cell data set with their Visium data set and identify, you know, map those cell populations that were previously identified with single cell and map them onto the Visium data. They were again able to confirm the expression of BSR1. So they, you know, could confirm that this was in fact present in the tissue. Not only could they identify it, they could also identify where it was being expressed. So, both of these papers are great reads. Great examples of how you can use FFPE, and we, as Flex continues, to be used, and as people start to use it more, we're going to have more and more publications. We have a lot of internal data sets that you can play around with, but I highly recommend looking around, at these publications. There's only a couple just because it's a new product and it takes a while to run experiments, as you know. But we're going to see a lot more publications in the future that are really going to help support the high sensitivity and the great results that you get from FFPE data. So to close, there's a lot of benefits of working with Flex. It's the most sensitive assay that we have, more so than 3'. In terms of sample compatibility, you can pretty much run whatever you need to run. You can either, if you have fresh or frozen tissues, you just fix it, or if you have FFPE, you can run that as well. Very simple, streamlined workflow, simple library construction step. It is an end-to-end solution. So we have Cell Ranger that can demultiplex your data if you're doing multiplexing. It's very intuitive software, so you can really analyze it, on your own. One of the most attractive things, of course, is the cost. You can achieve massive scale and some massive cost savings, especially when you do multiplexing, especially with the 16-plex, even with the 4-plex. No matter what you do, your sequencing burden is going to be halved. So it's going to be cheaper no matter what. Definitely the most flexible assay, and hence is, definitely, let me rephrase that. So definitely the most attractive part of this assay, and hence its name is the ultimate flexibility that you have. Just being able to fix and store your samples, run them whenever you want. You can batch your samples together. It's really great for time point studies. You can wait and collect them and then run them all together to remove any batch effects. Just increase your efficiency. Wait to run all 8 at once, rather than just 1 or 2 at a time. So really, Flex opens the door for new sample types, but it also opens the door for making this single cell workflow, which can be challenging, especially for the sample prep. It just makes it easier and more user-friendly. You're not on that time crunch. You're not worried about killing your cells. Fix all your cells, fix all your problems. It's a great solution. But to really drive the point home and just touch upon the introduction of this talk is our compatibility for FFPE across all 3 of our platforms. We really just talked about single cell Flex today. If you want a spatial context, you can also do Visium. Same exact probe sets, so you can integrate your data sets together. And now that we have Xenium, you get the highest resolution possible into those FFPE tissues of interest. Using a single assay alone is going to create those really rich, powerful data sets, where you're going to get extra power and more enhanced insights if you start, applying other assays together. We're really a one-stop shop for your FFPE solutions, and what's nice is that not only do you have consistent biochemistry between the 3 different platforms, you also get the full support for all 3 of our platforms. So if you start with single cell, go to spatial, your biochemistry is going to remain consistent, and you're still going to get that world-class 10x support, all the way from sample prep down to data analysis. So with that, that was the overall summary of our Flex assay. So I thank you for your time. Happy to take any questions. Yeah.
That was great and really exciting, encouraging. But I have a question about the probe set and custom probes.
Mm-hmm.
What if we wanted to look at coding RNAs or transposons? Those are not represented in the current set of probes.
That's correct.
Right?
Yeah. So we only do coding transcripts. You can do non-coding RNA. Just a couple of things you want to, and the techno describes all of this. There's just a couple of things you want to avoid, like long strings of repeats, because then you don't really know where it's going to bind. You want to avoid common SMDs. You don't want to have too many mismatches in your target. GC content is important as well. You want to make sure you're capturing targets with the appropriate GC content, so those annealing and denaturation temperatures. that it's going to work with our current protocol. Just a lot of different considerations. I think the main one being each probe targets like 50 nucleotides. We recommend 3 probe coverage for each gene for maximum sensitivity. You can just do 1. So if you have a really short transcript that doesn't have 3 ideal locations that you can, hybridize your probes to, you can just do 1. But a lot of considerations to take in mind, but we walk through, every step of the way.
Well, I'm interested in transposons, which are repetitive by nature. So
yeah, that might be tough because if it's a really, it's a really long repetitive sequence, you don't know like where along the sequence it's going to be binding. But let me dig into that more for you. The custom probe tech node is pretty new. So we're just starting to see people really implement it and use, hack the system and use it for their own work. Let me dig into that and see what other people have started to do. Yeah.
So for the papers that you showed so both the papers for the FFP samples that are nuclei extraction. So does that mean for FFP samples you're doing single nuclear sequencing or? So
our protocol is for whole cells. That's what's officially supported. If you want to do nuclei, you certainly can. It's just not going to be, you just follow the SN patho-seq protocol. I don't think it's going to hurt for either one. If you have the option, if you want to look at whole cells, I would just follow the 10x method because as soon as you deviate from our protocols, you don't get as much support. If you're just going to have to reference the publications. But if you follow our protocols, we'll have more insights because we've tested it in-house. We haven't tested SN patho-seq in-house. Really, I don't think there's a wrong way to go. Both of those, we've created really beautiful data sets with our FFP dissociation protocol, but SN patho-seq and that other publication also created really beautiful data sets. So either one really works. Yeah.
Is there a possibility to have focused gene search to look on the dispersion of several hundred genes?
Yeah, that's. Oh, wait, I'm sorry. I might have misheard you. Can you just repeat that?
So to look at fraction of the genes, not the whole transcriptome that you mark it, but
just do a subset?
Yeah.
There's no way to kind of just go into the probe panels and like pick out the genes you don't want. But with Xenium, we have pre-designed panels of a couple hundred. We're working to expand those numbers. So if there's only a couple of genes that you are really interested in, and you really want to get that high resolution, Xenium could be a really good solution. Because you can look within the individual cells. I didn't really talk about spatial today. That would be another discussion for another day. But again, we have data sets available online, so you can see what that data looks like. See how far you can really zoom in and see what kind of genes you can detect. But the short answer is, the panels are kind of as they are. They're really sensitive, so you're not wasting that many sequencing reads on those extra genes that you are interested in. You can computationally filter those out if you don't want them. It's easier to add on genes than to take them out.
I guess I'll just kind of follow up on the. So hi, I'm Anne. I'm your sales representative. We used to have a targeted assay where you could just target specific genes. In the end, not that many people purchased it. It just wasn't a particularly popular product, and so we chose not to continue using it. And so, as Caroline mentioned, if you're the targeted options like moving into spatial with Xenium is probably your best bet.
It's also pre-selected panels, not a custom panel.
Xenium has the option for both. So there are pre-designed panels that you can buy off the shelf, or you can do your own add-on. So if they're missing a couple of genes, you can add on up to 100 to the add-on panels. Or
Yeah,
you
can do up to 100 or fully custom 300 genes for really whatever you want to look at.
I also wanted to mention next week on the 19th over in Cambridge. I realize it's a trek. We are actually having our Spatial World Tour where we're going to be showcasing data and new information about the whole transcriptome with Visium as well as the Xenium targeted assay as well. So if you guys haven't registered, we'd love to see you there.
Free food.
What other questions do we have? Otherwise, I think the three of us we're going
Everyone's
to
hungry. Yeah.
we're going to stick around and we're available for more questions. I think you guys also had a session on talking about jobs in industry. Is that correct? No, am I making that up?
Can you talk a little more about making suspensions from frozen tissues?
From frozen?
Yeah, because that comes up.
Yeah. So are you talking about like